Okay, thanks the committee to invite me to present my work here. My topic today is theories and perspective. How big data and AI accelerate the discovery of a universal cancer principle from the non-coding RNA regime. And my topic contains four subjects over there. One is the crisis in cancer research. Let me check what of the data here. Check out the data published right now. A huge publication, like every year have 1,000 publication in 2024. Last year have more than 40,000 publication. It means every day have more than 100 publication associated with cancer mechanism, the molecular mechanism. I thought PubMed. And then I thought the Nobel Prize associated with the cancer, and then from the GTP, and then you see, and this have a Nobel Prize associated with the cancer, and then if you search more, have more like a gene regulation Nobel Prize have more of the cancer, but the cancer still cannot curable, right? So you see the data here, the publication count, and also the mortality, the cancer patient mortality. You see almost no relationship. How the trend go down, you have publication more, and then will go down mortality a little bit, but not so significant. So it means most of publication don't make sense, including Nobel Prize not make sense and kill the cancer. Actually, cannot kill the cancer. No way. Why cannot do that? Why is this crisis happen here? In the two part, one is the biology, and biology part is sample size limitation, because the all research right now limited the sample size. A cancer patient have million ODR, but there's research sample maybe focus on one type of cancer, and then or one group of cancer. There's cancer size limitation. The second, the traditional concept. Today's concept define the gene, and then model gene define the protein, and then like focus on the protein, like model publication on TP53. That's protein in cancer. The top one publication, if you search the publication website, you see top one is the TP53. Not only cancer, all the publication in PubMed, if you search the top one, the TP53. Why? Because that's a concept follow other people for traditionally. Actually, TP53 is not important in top one in cancer. Maybe not important in top of 300. I saw your data later. So actually, the bias, the traditional bias, is not actually the true as is a bias. Also, the tissue also the bias. Like use the different tissue to start the cancer. Actually, all the cancer, no matter what kind of tissue, is always outgrowing in the cell. We have similar mechanism, but always go to tissue. Also, why not go to the precise medicine, special one type of tissue, special one type of the gene, protein location, and mutation in the sample size. They're saying not a mechanism. In treatment, maybe have a little bit, but in the mechanism, nothing happened over there. Another bias come from the competition. Bias first from the conventional database. Conventional database like the gene code database. They do a lot of job. Oh, they are excellent, but they only define about 64,000 genes in this database, and then most of people, almost all the people, use this database, gene code database, to analyze data. So all the data come up is an unbiased method, like before have microwave unbiased, right now have sequence unbiased, but actually the analyzed data is biased. That is biased on this 64,000 genes. The actual gene in the human genome transcript in my bigger data, so how million of this kind of non-coding, all they are undefined, unrecognized by the gene code database. So actually this bias, and the algorithm also by the algorithm. Before we try to use unbiased algorithm, because system biology, what kind of mechanism try to unbiased actually bias, because they all the algorithm almost focus on this gene code database, in the unknown database, the protein database. So really biased, right? Biased, cannot see a whole picture. That's why the cancer research cannot move forward. Always go to that kind of bad cycle, come back again. Nobel Prize don't make sense. Like the one Nobel Prize say, oh, the mutation of the chromosome or loss of chromosome. My God, loss of chromosome can cause a lot of disease. And then how many cancer patients lost the chromosome? Loss of the chromosome is the consequences of the cancer, maybe after cancer and the loss of chromosome, or the loss of chromosome caused the cancer. Why? So most of the cancer it don't need loss of the chromosome, the baby, and then environmental factor change a little bit, and immune system change, and then cause the cancer will increase. Don't need a chromosome change. So all this research, Nobel Prize don't make sense for all the things. So how to make sense? Oh dear, how can get the data unbiased or for take a whole picture? So we move on to the bigger data discover. The bigger data right now have two databases I saw you here. One is the Cancer Genomic Atlas Program, we call TCGA database. That's the bigger database in the world for the cancer. Another database is called Sequence Read Archive, called SRA database. That database much bigger. It include all things. Every people publication, you submit the data over there, and then the database have millions of the RNA data, a lot of data over there. Okay, I use these two word biggest large data, and then to find out what the fundamental principle or the idea in that data. And then I simply put the cancer TCGA data and product, use the CCA, it called CCA called canonical correspondence analysis. And then CCA plot here have vector plot as a vector. Vector have two property. One is the strength, and another one is direction. It means the same direction, and then more longer the line and the more important. Okay, so you see here easily found out the alcohol more important than the cigarette, like smoking, the cigarettes. In the case, most people believe that smoking cause most of the lung cancer, cause that cancer, most important factor, but actually not. The alcohol is double important than the smoking. When I try to publish the paper, most people don't believe it. The review don't believe it. So after my publication, I will submit on website in early and publish in 2021. Right now, recently, you see a lot of people use the data to prove this is true. The alcohol most double important than smoking. It causes the cancer. So it's very powerful. And then right now you see a lot of people follow this idea of the publication, include the International Cancer Research Association and Harvard University published recently. And also to this point, alcohol most important. So before I found out this one, no one can say that. But after that, I follow idea. And then you see also here, is most of people saw the size, the cancer size, the tissue is important, but actually not contribute to overall that genomic size. A lot of the epidemiology variable maybe not cause that kind of genomic change. Most important, that's where higher variation the cancer cause, and no one knows what's the actually factor contribute this side. So that encouraged me and motivated me to do the research on this bigger data. So first of all, we need to define what kind of gene it is. How can measure all the variants in the genomic. So the traditional conventional gene is called, I have one block here, and most of that is the endogenous region. And then they define the protein only occupy 2% of human genome. It means 98% is unknown uncoding, and not so much understand their genomic. So their function in the genomic. So from the bigger data, I divide that genomic in 300 base pair window to count this kind of the RNA-seq data transcript. Unbind, I don't care what kind of gene it is. After after that, I remove this kind of the defined region of gene, already defined region, and then count the non-coding region to find out what exactly doing in the whole genome, and find out most of the human genome, almost 99%, 100% is transcript activate in some point. And so it's very important. However, hold the data is very complicated, no label, ODR, you can have no clue what kind of data it is in that XI database or PCA database, and no one knows what kind of thing. We are complicated, and then how can extract the information, use information for data? So I developed one algorithm called Phoenix to unbind, to understand what kind of gene regulation for this one. The algorithm idea of the sample just randomly split the data into M group, like M equal A here. I saw you for example. So it will find the gene B regulate A in all the the group, and then that's B and regulate A genes, the gene A, B regulate gene A, it might be true. Of course, that's process evaporate in n time after maybe 50 times regulate. It will still found the B and regulate A, and then it is true means B regulate A. So this algorithm to find out whatever data it is, we can find out the true interaction. See here, I put this algorithm to find the two largest database in the world, and then you find out the interesting pattern here. One is the normal condition. You see, there's a light green is the protein, and then the less and the non-coding. And then in the cancer, wherever in the protein region, you see the density very, very low. And then here the density very high. It means the protein interaction not important as in the cancer actually. Is most of that non-coding RNA, this four region. You will see the state data here, statistics of that graph. In the normal condition, is the inducer, most of the cancer, most of the in the protein coding genes in the inducer in normal condition, and 60% top of 300, 60% of that is a protein. That's in normal condition, and then that inducer target also the protein, most of that. However, the cancer, not protein here, all the pseudogene and antisense RNA, the long non-coding RNA, or no protein in inducer in top of 300. So that's why I say the TP53 is not important in 300, and maybe not important in 1,000. But other people still publish a huge paper right now. You see that. The Nature Science paper still publish a lot of TP53. The research actually don't make sense in the cancer research, but they publish. So whoever the inducer, the targeted protein, this is why we say the protein important, not important, but can observe. We can find a lot of protein change in the cancer because the cancer induces the long non-coding RNA and induce the protein. So the fundamental mechanism is the non-coding RNA. We saw the protein change, but the protein change caused by the non-coding RNA. The long non-coding RNA most important. The fundamental mechanism, the non-coding RNA, not protein. Protein, one kind of phenotype. We see the protein, but actually they're not important. They're not fundamental mechanism. Wherever you talk about it, wherever publish cancer research, Nobel Prize doesn't matter because they are not fundamental mechanism. That's why we have this crisis over there. And then you see more detail over there how the non-coding RNA regulate the protein. And then in the normal condition, you see the non-coding RNA, they regulate the whole genome across the chromosome. All chromosome, it cannot separate. However, in the cancer condition, the chromosome always separate one by one circle, the chromosome there. It means the long non-coding RNA, the non-coding RNA regulate the protein in the stem chromosome, and that very limited number the cross regulate or in the cancer type. So the mechanism, the model should be something here. Conventional model is called protein, regular protein, and then however the tool found out from the bigger data model actually the non-coding RNA regulate the protein in the cancer in 1 million base pair. That's where the size regulation and in that one. So take a home message is the non-coding RNA regulate the protein cause the cancer. That's the take home message. Of course, if you have this idea and then have this principle, and then easily use the AI, use the non-coding RNA together, and then you can find out that the non-coding RNA and AI can separate the protein level. Can separate the protein in very high accuracy, like 96%. If the individual cancer type over 99%, 100%, no problem. 1 or 2 over there. Of course, there's AI model much better than traditional model, and then so. Overall, that's the kind of non-coding RNA found for bigger data will have more advantage and then more true in cancer. So, in that means we can easily use the principle and use this idea to develop the way develop the DLP to treat a patient. Maybe now, so right now we still have a challenge over there. The first challenge is the non-coding RNA functional system. We don't know the functional system right now, non-coding RNA. Traditionally, we still use the protein functional system to understand the non-coding RNA, like in the conventional model, and then we still use polymer 2 or polymer 1, 2, 3, that's things, and then say the protein regulate the non-coding RNA initiation, the transcript. However, in the bigger data, I found out that actually that non-coding RNA, the regulate by the non-coding RNA, not protein. Most importantly, the factor is not polymer 2. This is unknown factor. No one knows in this unknown factor in the world except me. I computationally understand and I know this factor, but I have no experiment prove it, so I have no published this factor. So just believe me, this non-coding RNA functional system is not the conventional protein system. This is a very new system. This system is very, very important because they will account for 98% of the human genome. And this 98% of genome not only accounted for the cancer, they also account for the another disease, infection disease, also the environmental change, something like that morning you drink coffee, tomorrow you maybe drink tea. That's the time of thing. The gene expression is different. The gene expression different. What caused the gene expression different? Because that's kind of non-coding RNA functional system change. So you don't need change every DNA sequence. You don't need change anything. The non-coding RNA change expression activation a little bit different, can regulate that thing. So that's a non-coding RNA functional system extremely important, more important than most of that kind of the Nobel Prize, that things, the protein system in the human genome. It causes 98% of human genome, but no one knows this function system. So what is a very bigger challenge over there. We have not understand it. The second challenge we have not animal model to start a non-coding RNA. Most of people right now use the mice model to start the protein system, try to start the non-coding RNA. Actually, it's not correct. So you see here the data, and the red color is a human, and then the blue or green color is the mouse. You see the range, and then the human have much more define the non-coding RNA. That's only defined by the gene code data. And actually, they are much more longer, and that's a non-coding RNA over there. And then your protein, of course, very similar, and then also human a little longer. However, the gene number, you see, that in the mice have more gene in the protein genes than the human. That's why if we use mice to start the protein, of course, might be is okay, and maybe more than 80% or 85% of human or mice, they kind of protein is homologous, but in the non-coding RNA, quite different story. Than the human have much more the genes than the mice. Only for the gene code data. For my data, of course, much more, millions of that kind of non-coding RNA, and not so much mice can non-coding RNA can find. So absolutely not appropriate to use the mice model to start a non-coding RNA. Absolutely nonsense over there. So in further challenge is the idea, the concept of that kind of study system. In the protein functional system, we try to use the conservative pattern, like if such energy, and then you found a lot of animals like the mice or the dog, wherever a lot of even jellyfish have homologous in the protein system, and see all the conservative system. However, in the non-coding RNA, no, yeah, they're quite different. For example, you will see 4 motifs here. I use an example. The left 2 motif is associated with the lifespan, the longest living. Human can live more than 120 years. Human and mice only live less than 5 years, most of the 2 years. And then you see this, the left 2, that's motif like a CGTATA and GGTCCG. This is motif. It will greatly get this motif. The mice have very limited of this motif, while the human have huge of this motif. Evolutionary, the dynamically evolution from that, not conservators. However, in this the left 2 motif, and then they lost after live longer animal, and then they will lost that motif, and then so this two motif, actually the short-lived motif, and then there's a long motif, and then the long-lived motif. Of course, you can compare to the cancer also. The cancer live longer, and then have some motif, and then will evolutionally increase some motifs from the cancer. If no cancer, some motif will limited. And then after evolutionary dynamically increase some raw material. Actually, that's true. Right now, some database called, they will call some longer living material genes as the oncogene. They call it cancer gene. Actually, this gene is very important for human life. I will show you the data later. So the new concept we need to identify these things. So the new concept required for microRNA study. Then the fundamental. So the fundamental question is, what is the human genome? Traditionally, we say that the gene information of organism is the human genome. Gene information, of course, the biology people maybe understand what kind of gene information it is, but the computation people don't know what kind of genome, the gene information. So have no way to compute what genetic information. Genetic information maybe need to define the gene, and then divide the gene is biased, so it not make sense in the computation. No one knows what's the meaning here. And then also the method to study that kind of genome is not appropriate for study the non-coding RNA. They first of all, they ignore the major difference, like a P sequence here, Q sequence, 2 sequence, or the A, and then you see almost the same. So traditionally we use the alignment. We say, oh, the 2 sequences the same, 100% alignment. Actually not, because Q gene has 2 AA, they are longer than this one. That's than P. So the 2 AA, of course, they can code a different thing. So the traditional alignment just found out one kind of mutation in the middle, like A mutation to G something. Oh, how mutation ODR cause the difference? Actually not. So it caused the difference because the Q have more AA ODR. So you found the kind of mutation, point mutation, or the group of mutation don't make biology any sense. So maybe 20 years ago, a lot of publication in Science Natural caused the SNP profile, called little point mutation and cause the phenotype. Actually, so far no one single SNP profile associated with the phenotype. So all wrong. This kind of the traditional idea of publication wrong, and of course the huge publication at this time. But right now you can see a lot of general publication of SNP or things. Also, that kind of the alignment is not good for data. Cannot compute. So how can define compute the genomic? A genomic here I define here that a genomic is the precisely older sequence of 4 nucleotide ATGC. Okay, so it means it precisely older sequence of these 2 ATGC. So ATGC can different order, in a different order, and then it will change that kind of sequence. So okay, that kind of genomic sequence can define to the spectrum of the molecule, like this molecule of course can derive from the complete mutation of 4 nucleotide ATGC. This description maybe not easy to understand for biology, but actually it precisely can compute. You can compute for the computer science for that make a meaningful. I put a sample here for biology understand. So if the one base pair and motif can of course have four the sequence ATGC sequence here, and then if one base pair and then have four motif, two base pair of course has 16 motif. It will complete permutation. Of course, 3 motif of 65, and then 4 motif of course 255, so on, so on. A different motif can compute it, and then I use one of the matrix, and then you found the sample 1, and then in the motif A, and then whole different from the original. For like for this P and Q, and then the different two side two AA over there. So the different calculate the distance, the evolution distance from the original reference, and then you find a matrix, and one is the motif longer, and then another one is sample, and then this matrix of course is easy to compute for the computation and AI and bigger data. Then make the bigger data computable, and then AI can involve it. This is why a publication use the AI, and then that's algorithm to start a couple millions of the SARS-CoV-19 sequence over there, and then. So in that algorithm, we can find, of course, the motif like I saw you before. This kind of long motif in the human and the mice comparable, and then you see that motif much more in human, and then less in the mice in the long. However, in the short lifespan motif, and then the human have less, and then the mice are long. This provide additional line of evidence that the mice is not a good model to start the human non-coding RNA, because the motif different, the gene is different. And if you map this motif go to the gene, and then you found more interesting, like the SC15 gene, and then the genes distribution in the human tissue, and in the neural system, and animal like in the human or various system, and in the mature system, in the producer, in the reproducer system, and the neural system. So the longer motif, a longer gene, and then in a woman, and then also the tissue. So the reproduction in the human, this is why the woman have long life, because the woman carry more of this long liver motif than the man. So the why the woman live longer than the man, and also that's a gene also called the anchor genes in traditional publication. Actually, no. If you use the target this gene, and then maybe let the man, the woman, it was shorter, because they're very important for the long living. So it's misunderstanding. This is very important for the long living, for the living age, the longer ways. So not the cancer gene, actually the long living genes. So if you understand that one, and then you go to the database to check out my data, to check out what's going on, to start this in the cancer. Of course, for perspective, Okay, my time's limited over there. For right now, how can use this AI algorithm? Okay, of course, the AI also use save attention and like AI like language model like ChatGPT or DeepSeek something. They kind of model come out. For the language, of course, is okay. The language, very simple actually. Like my presentation format is what kind of format is it? PPT. Of course, my presentation not PPT. It's ODP, PDF. Very limited the format in the language. However, the human genome is different, and one gene can cause hundreds of phenotype over there. So that kind of language model is not exactly fit for the genome research, actually human genome research. Of course, there are a lot of people publishing use that kind of model. They can publish a lot of natural paper, science paper, but actually not make a lot of difference. However, that kind of AI idea is very important, very a good idea. Like an AI can communication to 3 sections of multiple sections. We can separate this kind of data, bigger data, into different sections using AI to learn it, and then can communication together. In the traditionally, a human, if you put a group of the scientists together to talk, and then they cannot understand each other, and then cannot understand it, cannot communication. By AI, they can. So we can correct the data in different sections. Of course, the data correction not only limited the genomic, maybe the clinical data also important. If we can put all data together to split it into different sections to learn it, of course, we can find a lot of interesting things over there. For overall, we use AI and big data. Finally, we need found the universal principle somewhere. Of course, it will go up to we can find the physical law and the biology. A physical law, biology, of course, is not like not a sample mechanism. A lot of cancer mechanism actually not the law. The law maybe not contributed by the genome. If you started the biology law, the sample law, maybe found more fundamental element than the genome. The genome can evolution by the law very simply, like the Newton fourth law, the second law, cause F equal ma, this kind of physical law. And then we need more talented people and to start that kind of sample law, and need somebody like Isaac Newton or Nikola Tesla, that kind of talented people to start it with the physics and mathematics to start it. That's kind of law. And then, of course, why not we starting the principle? Of course, my talk, all the data not so far away from here. Actually, you can go to that's kind of database to find out more interesting data over there, like in the cancer database for the bigger data, and then you can search like the Penton one super gene, and then you found the network here. I say Penton gene actually more interestingly, and then is the super gene actually that regulate the Penton and contribute more Penton. So right now, most of people started Penton is more important than the another gene, like Penton, of course, more important than TP53 in the cancer. But Penton actually the regulate by by their pseudogene, and then the pseudogene also regulated by their antisense, their network over there. I'll call it, if you can start that PEN10 network, and then they contribute more than most of science natural paper. I will call it. You can search PEN10 and PEN1 if you have more interesting. And then you can find you find more of this, and then you see a lot of all that's kind of all the interaction over there, and then there's for the database, for the bigger database come, and you'll be able to say all the protein regulate the protein, but the oral protein actually most all the time regulated by the pseudogenes network here. You see the pseudogenes over there. So the pseudogene or the non-coding RNA are more important than the protein over there. We found the protein phenotype change something in the cancer, but actually the fundamental mechanism is a non-coding RNA. So what I talk here today, and then interesting, and then you go to the website to check out, and then thank you. I'll be happy to answer your any question.